Good morning, everyone. Welcome to the Department of Health and Human Services. We want to thank the families affected by rare diseases who traveled this weekend to be here to celebrate this special announcement with us today, and thank you for bracing with us with the weather and the fire alarm. We appreciate it. Thank you also to the doctors and scientists who did the same, and thank you to the members of the media in advance. So I would now like to welcome our Health and Human Services Secretary, Robert F. Kennedy, Jr. Thank you.
Thank you all for coming, and I'm sorry for the interruption. I've told them not to schedule stuff on Mondays because it seems to be a pattern. This summer I met Kyle and Nicole Muldoon, the parents of baby KJ. Baby KJ from Clifton Heights, Pennsylvania became the first patient in the world to receive customized CRISPR gene editing therapy for a devastating rare genetic disorder called CPS1 deficiency. The doctors diagnosed him shortly after his birth in August of 2024. His condition was life-threatening. Kyle and Nicole refused to accept that diagnosis as his destiny. They partnered with physicians at Children's Hospital of Philadelphia and Penn Medicine and helped pioneer a gene editing therapy built specially for their son. After 307 days in the hospital, baby KJ came home. That's not just a recovery, it is a medical miracle. When I heard KJ's story, I directed HHS and the FDA to move quickly to ensure that this breakthrough technology would not remain a one-time medical miracle. Every child facing rare genetic diseases deserves the same kind of treatment that KJ got. Please join me in recognizing two of the physicians who made that breakthrough possible, Dr. Kiran Musunuru and Dr. Rebecca Ahrens-Nicholas from Children's Hospital in Philadelphia. I also want to recognize some of the congressional leaders who helped us to make this happen, who've driven this agenda for a long time on Capitol Hill. One of them, Senator Roger Marshall from Kansas, who's a physician, and who I consider not only a friend, but a partner in all of the MAHA agenda. And then Nancy Harshberger, the congresswoman from Tennessee, who's also a pharmacist. And thank you so much for your activism on this issue and your very strong advocacy. And then Nick Begich, the congressman, the sole congressman from Alaska, who's helped drive this issue, and his wonderful wife, Darna, who is also a pharmacist from Alaska. Thank you for being here today, and thank you for your visionary leadership in making these kind of treatments available for every American. And to the physicians and researchers, advocates, and families in this room, your persistence brought us to this moment. Today, we turn that persistence into policy. The FDA releases draft guidance establishing a new pathway to approval individualized therapy for rare genetic disorders. And we're calling this the plausible mechanism framework. This framework aligns regulation with biology. For ultra-rare conditions, randomized controlled trials are often and mostly always just not feasible. Under the framework that we're announcing today, one well-controlled clinical investigation supported by confirmatory evidence can support approval. For decades, families heard the same thing. There are not enough patients, the approval will take too long, you just have to wait for the science to catch up with your child. That ends today. A disease with 100 distinct mutations in the same gene will no longer require 100 clinical trials. When biology is clear and the science is sound, we will evaluate therapies based upon strong evidence and not arbitrary barriers. Individualized medicine is no longer theoretical. It's a child breathing on their own. It's a mother sleeping through the night. It's a family planning for a future instead of fearing one. To the doctors here, bring us rigorous science, define clear endpoints, collect strong data, move with discipline and speed, and we will move with you. To the parents here, you endure delay from a system not built for diseases this rare. We are ending that delay. You should never have had to fight the government while fighting for your child's life. We will insist on transparency. We will require long-term safety monitoring, and we will act with urgency. Medicine advances one patient at a time, and regulation has to keep pace. This reform builds on other decisive actions that HHS has taken for Americans with rare diseases. A few months ago, HHS added rare diseases, Duchenne muscular dystrophy, and metachromatic leukodystrophy to the recommended universal screening pattern for newborns. That decision allows physicians to identify, diagnose, and begin treatment sooner when the time matters the most. The Centers for Medicaid and Medicare Services Cell and Gene Therapy Access Model expands access to life-saving therapies by securing discounts and outcome-based rebates for states with treatments that deliver results. This is about more than policy, it's about people. You're going to hear today from Judy Stecker about her son Wheeler, the youngest CLN3 patient ever diagnosed. Julie Vitarella is also with us today. In 2018, the first Trump administration cleared the first medicine made for a single patient, Julia's 8-year-old daughter, Mila. Her therapy, Milacen, opened up a new frontier. Mila's disease eventually progressed, and in 2021, her big spirit left her body. What Julia began as a race to save her own daughter became a movement to break the regulatory barriers for children with fatal genetic diseases. We also recognize Gwen Traficant. When her husband Jim was diagnosed with a rare liver disease, she stood behind him through 2 grueling treatments. Years later, her daughter Ashley faced the same diagnosis and underwent 2 more. Their son Jeremy became a living donor to save his sister's life. Today, Gwen chairs the Virginia Rare Disease Council and guides families through a system that she knows firsthand. We stand now at an edge of a new era. Genome editing, RNA therapies, mutation-specific medicines designed for individual patients. Innovation without accountability destroys trust. Regulation without flexibility destroys hope. We reject both extremes. We choose innovation and accountability. We choose speed and safety. We choose science and compassion. Together, we will accelerate cures, empower physicians, heal patients, fuel innovation, and make America healthy again. I now welcome FDA Commissioner Marty Makary. Marty understands that innovation without accountability destroys trust, and that regulation must keep pace with science. Under his leadership, the FDA is doing both. Marty has been, in my view, the most important, the most effective FDA leader in history, and I'm very, very glad to have him as a friend and a partner. Thank you, Marty, and welcome.
Thank you, Mr. Secretary. It's so great to be here and to reconnect with so many from the rare disease community, members of Congress who have been waving a flag in the air, advocating for this issue. It turns out rare diseases are pretty common. 30 million Americans have a rare disease. That's 1 in 11 people in our country. And for too long, they have been underappreciated, under-recognized. The research has been underfunded because our system is built for common diseases, not for rare ones. And I've taken care of many patients with rare diseases. And I'll tell you, it's hard. It's hard to look somebody in the eyes and say there's nothing out there that we know of that we can offer you. It has an impact on your soul. They don't understand that the market size is too small and it doesn't incentivize companies. It doesn't make sense to people, nor should it. When you're in that situation at the bedside, and I've been there hundreds of times, you ask yourself, is there anything out there or anything humanly possible that we can do differently? And that is the perspective that I bring to the FDA. When people say, why are you doing so much stuff? That's why, because they have been waiting for too long without satisfying answers for many of these conditions. Historically, rare diseases have been at the FDA an afterthought. Rigorous, elaborate standards so high that they're just not practical for many of the therapies for rare diseases. But today, treatments for rare diseases have powerful incentives, expedited reviews, tax credits for clinical trials, extended market exclusivity, and a powerful incentive, if the drug is approved, a pot of gold of $100 to $200 million in the form of a transferable priority voucher. So we've come a long way, thanks to many of you who have been championing these ideas. Today, the majority of drugs we approve at the FDA are for rare diseases. It is the majority of our resources. In December, FDA's Center for Biologic Evaluation and Research hit a record number of approvals. But we can't be satisfied. We can't let up. That's why we're pressing ahead. Last month, we announced new manufacturing flexibilities for cell and gene therapy, cut the red tape and speed up many of these potential cures. You don't have to run 3 manufacturing batches routinely anymore, thanks to this announcement. We've removed the long, onerous requirements for follow-up for people who get CAR T therapy. We don't want investors scared away because of the high cost. We want investors to see the bright promise of rare disease therapies. We announced last month at the JP Morgan Healthcare Conference that we're embracing fully Bayesian statistics that are ideal for small populations and pull in data from other trials to help make conclusions. That's not just a p-value of 0.05 above or below. It's not a frequentist dichotomous approach. We can learn from individual patients. We've approved every single compassionate use right to try authorization that has come across our desk. And we believe in both the spirit and letter of right to try. And thanks to Congress a few weeks ago, they passed, and we were on the Hill actively pushing for this, the Pediatric Priority Voucher Program. So it is renewed and signed into law just 2.5 weeks ago by President Trump. We're going to keep going. Today, we're announcing new guidance at the FDA on the plausible mechanism pathway. What is a plausible mechanism pathway? It's common sense. For the first time, the FDA is issuing guidance where giving an ultra-rare disease therapy, giving drug developers of ultra-rare disease therapies a path to accelerated or traditional approval based on the experience of individuals. We can learn from individuals. I have learned from individual patients of mine. And when you see something amazing right in front of your eyes, that is a type of evidence. It's time that the FDA recognizes it. So I want to thank all of you who have been pushing for this, and I want to thank Tracy Beth Hogue, who leads our center called CEDAR, and she's going to come up now and share a little bit with you. Thank you, Tracy.
Hi, everyone. Thank you, Dr. McCary. I'm Tracy Beth Hogue. I'm the acting director of CEDAR, the Center for Drug Evaluation and Research. And today is a very exciting day for patients living with ultra-rare diseases and the community, all of you, and across the world, the people that support them and want to see effective cures and treatments for these patients who it's been a struggle to find effective treatments and cures for in the past. But I want to let you know that during the fire drill that I think it was not a real fire. I'm still not sure. I didn't see any flames, but that I was discussing with two of the researchers here who have been foundational in the plausible mechanism framework, Dr. Musunuru and Dr. Ahrens, Nicholas, how we are going to operationalize the plausible mechanism pathway at the FDA. They are the scientists who are behind the treatment of baby KJ, who inspired the plausible mechanism pathway, as Secretary Kennedy just explained to you. So what is the plausible mechanism pathway? The plausible mechanism pathway is a way for treatments to get a special path to approval that uses the same standards for FDA approval as we've already used, as we've always used. But these treatments, they treat a disease with a specific molecular or cellular basis for the disease, with a treatment that directly targets this molecular or cellular or genetic basis. And then they are able to demonstrate evidence with this treatment that they've been able to alter the basis of this disease and show a clinical response or some sort of biochemical response that is different from that we would normally see with the natural history of these diseases. And then with these ultra-rare diseases, we can look at them not just as one individual disease, but as these researchers here and other researchers have been able to show that we'll be able to look at these as a collection of diseases in potentially one clinical trial, but that because they are similar treatments, be it genetic editing or antisense oligonucleotide treatments, as we would see in my Center for Drug Evaluation and Research, where they use a similar mechanism but treat different diseases, and thereby we can treat a larger patient population. And so this really epitomizes what individualized medicine is, and it's so exciting to see this happen. And what I was part of this effort in CEDAR and CBER, a cross-center effort to write this plausible mechanism framework draft guidance. And it was very exciting to see our centers come together and the enthusiasm that everyone had for this, because I think we sensed together that this is going to be a transformational draft guidance in the sense that it's going to inspire industry to start developing these treatments in a way that we haven't seen before, and that we all feel, and I know that the researchers up here today feel similarly, that the FDA is likely going to be overwhelmed with applications for these treatments for these rare diseases, and that's wonderful and exciting. And you know, we're going to need to be creative in the way that we meet this new demand. But this is such great news for patients and the family members and communities living with patients with rare diseases. And so you've already heard a bit about baby KJ, who really inspired this plausible mechanism pathway. And so now I'm very excited to introduce you to one of the researchers who was the first author of the paper in the New England Journal of Medicine article outlining the treatment of baby KJ. And I highly recommend that you read that as well as the plausible mechanism framework in the New England Journal to better understand this topic. And so the researcher that I'm going to introduce you to is Dr. Kiran Musunuru from the University of Pennsylvania. And he is a distinguished professor at the University of Pennsylvania and a practicing cardiologist, and has won multiple awards. And it's such an honor to meet him and to introduce him, and he is going to tell you a bit more about the plausible mechanism framework and genetic editing for ultra-rare diseases. Thank you, Dr. Musunuru.
Thank you so much for the very generous introduction, Dr. Hogue. It's a real privilege for me to be here to speak to all of you on this occasion. I should note that I'm a physician scientist and a practicing cardiologist, so even though this is a Rare Disease Day event, I want to start by talking about heart disease, the leading cause of death in the US. To tackle that threat, my team created a genetic therapy to turn off a cholesterol gene in the liver, permanently reduce cholesterol levels, and give lifelong protection against heart disease. And this therapy is now showing signs of success in clinical trials. Now, the beauty of this kind of therapy is that it has a small component that acts like a GPS. Switch the address in that GPS, and the therapy will go to another gene and make a desired change. Otherwise, it's pretty much the same therapy. So as you've heard, in the summer of 2024, a baby boy named KJ was born at the hospital of the University of Pennsylvania, where I work. He immediately became deathly ill from an ultra-rare disease called CPS1 deficiency, caused by a misspelling unique to him in one of his genes in his liver. He was so close to death that there was actually serious discussions about transitioning him to comfort care and allowing him to pass away peacefully. But his parents rallied. He was moved across the street to the Children's Hospital of Philadelphia to receive life-saving treatment. And with funding from the NIH, specifically the Somatic Cell Genome Editing Program, and working closely with the FDA, we were able to take that same heart disease genetic therapy, just change the GPS address to point the therapy to KJ's broken gene, correct his unique misspelling, turn the broken gene in his liver back on, and make him healthier. Although this was not a cure, he is now thriving, growing well, full of insatiable curiosity and unbridled joy. And we realized we could do this over and over again, individualizing the therapy to many patients with liver-centered diseases by just changing the GPS address again and again, correcting each person's misspelling. And so we've turned this therapy into a whole platform, again with funding from the NIH, working closely with the FDA, that will position us to eventually launch a single clinical trial for a whole set of liver-centered diseases, the urea cycle disorders. We'll be continuing our work with the FDA using the plausible mechanism framework that's being published today, with a goal of getting to an approval of this platform as efficiently as possible and getting individualized treatments for the urea cycle disorders to all the patients in the US who could benefit from them. But that's just the first step. We intend to expand this work to many other liver-centered diseases. And as we go forward, we'll share our learnings with everyone in the hope that when scientists develop technologies to get this kind of therapy to many organs besides the liver, it will allow doctors to treat many, many rare diseases by getting at their root causes. It will be a whole new medical subspecialty, interventional genetics, and the goal will be to fix things early and give the gift of life to the tens of millions of Americans who collectively suffer from rare diseases. They deserve nothing less. And now it's my immense, thank you. And now it's my immense pleasure to introduce my good friend and colleague, Dr. Rebecca Ahrens-Nicholas of the Children's Hospital of Philadelphia. She's a physician scientist like me. She co-led the development of KJ's therapy with me. We were equal partners. I'd actually give her a little more of the credit. She's an extraordinary pediatrician and metabolic disease expert who is part of KJ's care team. I'm so thankful to God every single day that I have the privilege of working with her. Here's Becca.
Thank you, and thank you for the kind introduction. And I want to express my gratitude to everyone for allowing me to be here today and participate in this important event. Every day, I have the privilege of caring for patients with rare diseases and leading a team of scientists in my laboratory in collaboration with Kieran's group and others to try and find solutions to help them. Unfortunately, our rare disease communities face real uphill battles when it comes to developing new therapies. Small patient populations and limited natural history data can make trials really hard to run. Add regulatory uncertainty, high per-patient development costs, and uneven access to diagnostic testing and expert clinical care, and it's really easy to see why promising science too often stalls before it reaches patients. The biggest hurdle, though, is moving beyond a first-in-human trial to regulatory approval. Getting past that gap is what allows treatments to become commercially available, reimbursed, and scaled, so all patients who need them can access them, not just those in a single small study. Since our work developing KJ's experimental therapeutic was published, Kieran and I have received thousands of requests from desperate families asking for our help. As a scientist, a physician, and a mom, this is heartbreaking. We really need to do better. This is why clear, pragmatic, consistent regulatory guidance is essential. The types of individualized genetic therapies that we're trying to develop simply do not fit in the traditional model of drug development. This is true for so many treatments for rare and ultra-rare diseases. And we need these innovative regulatory frameworks that will allow us to efficiently evaluate if these treatments are safe and if they work. However, I do want to raise a challenge to all of us who hope to employ these novel frameworks. With this opportunity will come great responsibility. When developing therapies using this framework, every single piece of data from every patient is priceless. We already know this in the world of rare diseases, but it will be even more true now. And I hope that we can all proceed with radical transparency and share all that we learn, both the triumphs and the struggles, as we build this field of interventional genetics. It's really the only way we will truly honor our rare disease patients. It is an amazing time in the history of science and medicine. Due to our robust tradition of federally funded science, we are the global leaders in designing transformational, cutting-edge treatments that will make our population healthier. And I'm really excited to see what comes next. And now, I'm honored to introduce Judy Stecker, who can explain firsthand why innovative frameworks like the one announced today are so essential. In addition to her history of service here at HHS, she's done a heroic job advocating for her son, Wheeler, a rare disease warrior. Thank you.
Thank you, Dr. Ahrens-Nicholas, for that kind introduction. And I want to thank Secretary Kennedy, Commissioner McCary, Dr. Hogue, and the department for inviting me to share my story. As we kick off Rare Disease Week, I also want to recognize all the rare advocates and families, including my own here today. Court, Wheeler, Reed, Mana, and Papa, you all make the rare disease community what it is. It was your advocacy and your voices that gave me strength on my darkest day, Diagnosis Day, May 29th, 2019. On that day, I was told my son Wheeler, just 4 weeks old, had CLN3 juvenile bat disease, a rare genetic disease that will steal his vision, memory, mobility, and ultimately his life. Unlike many rare families, we didn't have the diagnostic odyssey so many endure to figure out what is happening to their child. Years earlier, my husband Courtney and I learned we were carriers of this cruel disease. We spoke to families, countless experts, pastors, and priests. We prayed, and we decided to explore IVF with pre-implantation genetic diagnosis. After multiple miscarriages and failed cycles, we were told it was unlikely, if not impossible, that we could conceive. But God has a plan, and He made a way. I ended up serving in the first Trump administration here at HHS, and shortly into that role, against all odds and all efforts to avoid it, I became pregnant. We knew the risk, but we had faith. But on that May day, our lives were forever changed, and we began the journey that rare families face. Not enough data, not enough treatments, not enough help, and not enough hope. I felt helpless. What could I do? I reached out to colleagues at HHS, and soon our new family was at the NIH, joining an existing natural history study, providing critical data to better understand disease progression and perhaps one day help prove a therapy's efficacy. Treatments, however, would prove more difficult. I learned that the traditional drug development pathway, requiring multi-million-dollar trials spanning several years for drugs that treat the masses, were simply unfeasible for rare diseases like Wheeler's. To reach the promise of modern science, families like mine have had to build their own path until today. Through sheer will and desperation, they raised millions of dollars hosting bake sales and golf tournaments, finding scientists willing to develop a therapy, convince an institution to manufacture and administer it. While this is tremendous, it puts incredible pressure on families who are already juggling the challenges of a terminally ill child. And it only helps one child, one mutation, and one desperate family at a time. This is not a sustainable path. It is a series of individual acts of heroism by parents, doctors, and scientists. Parents like Julia Vitarello, a fellow Batten disease mom who just a year before Wheeler was born worked with doctors, scientists, and the FDA to develop a highly precise genetic medicine for her daughter Mila, who had CLN7. Mila became the first person in the world to receive an individualized medicine. Through tears, I shared our story, and Julia described the Herculean effort to get Mila treated, connected me with experts, and shared her hope and vision that what was possible for Mila could someday be a path possible for millions, including Wheeler. Years passed, but firsts build on each other, and in early 2025, I read about baby KJ, the first person in the world to receive an individualized CRISPR therapy. Families like mine have watched these scientific breakthroughs outpace our ability to get life-saving treatments to our children because regulations have simply not kept pace. But today, that is beginning to change. Today's announcement, the Plausible Mechanism Framework, recognizes the unique nature and enormous opportunity of individualized therapies to address the health crisis of severe and life-limiting genetic disease, and that an innovative process can, in and of itself, potentially be a product. This framework creates a commercially viable path to deliver hope, not one child at a time, but at scale, and lead the world in genetic medicine. Shortly after my son was diagnosed, a senior FDA career official, Lauren Silvas, came to visit me. She held Wheeler in her arms and said to me, "There always has to be a first, and there is no reason it can't be him." Mila is a first. KJ is a first. This administration, this president, this secretary, this commissioner have proven that they have the courage and commitment to make firsts happen. So to all those here and listening, government officials, researchers, payers and providers in the biotech industry, there are 30 million possible firsts in this country. 15 million of them are children, and one of them is mine. We need you all to work together to take today's guidance and translate it into getting individualized treatments to patients. They are waiting, and for many, time is running out. So let's get to work. Thank you.
Thank you so much to all of our incredible speakers. We will now open up for Q&A, and I ask that you keep the questions to the topic of today, and that you say your full name and outlet when you are called on. We have our girls here with the microphones, if you could speak into those as well. So we can get started. Yeah, go ahead. Right here.
Thanks. Zach Brennan from Endpoints News. I was curious, can you explain this seeming contradiction where you're embracing genome editing and you're embracing CRISPR-based systems, but you're not embracing mRNA vaccines? Is there a contradiction there?
Well, I think that would be quite a stretch to say we're not embracing mRNA vaccines. We have we've approved 2 mRNA vaccines at the beginning of my time. I would love to see the data on mRNA helping patients with cancer in a number of conditions. Our job is to review applications that come in the door, and if the data supports the claims that want to be made, then we will approve that product. So we have a very clear mandate from Congress, what's safe and effective, and so that's our task. Now, there was funding for mRNA technology at HHS. That funding was pivoted to other causes, and I'll tell you why. It's not because we don't believe in mRNA technology. It's because the companies that made mRNA vaccines made over $50 billion. They can fund their own research. It doesn't have to be on the backs of American taxpayers. We should be funding research that is otherwise not getting funded, that has no money for funding, such as funding for bespoke conditions and rare diseases, conditions that have been in the blind spots of modern medicine, and that's why that funding was pivoted. So it has nothing to do with the view of this is a promising or this is not a promising technology. Our job as regulators is to be a referee, and we just want to see good science. Thank you.
Thank you. Yes, right here, in the green.
Thank you. Good morning. My name's Kim Higbee. I'm a rare disease mom, and also I represent the Hope for PDCD Foundation. And I'm just curious how this relates to like diseases and therapeutics that are already in the regulatory pathway right now. Like specifically, our drug just got a CRL that we are working for right now. And is it only limited to like gene editing, or is this also for other therapies?
Hi, thank you for that great question. No, this is not, this pathway is not limited to only gene editing. We actually outline in the draft guidance how this will apply to, we specifically list antisense oligonucleotides, but leave it open to other types of treatments. And I'll give the example of a recent approval that we had in CEDAR for Zycubo, which addresses an underlying issue of copper absorption in children with Menkes disease, which is a genetic disease. And the manufacturers were able to show increased survival compared to the well-known natural history of the disease without a randomized control trial in a relatively small number of patients. And this is the type of evidence that we're going to be looking for with the plausible mechanism pathway. When you're able to show with a limited number of patients increased survival, better quality of life, better laboratory outcomes, which we've never seen before in the history of the disease, like with baby KJ ammonia levels decreasing by nearly 50%, and the baby being able to live in an outpatient setting an essentially normal life, which is inconsistent with the natural history of the disease that we've seen before. This is overwhelmingly compelling evidence of efficacy, and we do see these with rare diseases, and this is things that we have seen in the past and what we'll be looking for with the plausible mechanism. Now, with the plausible mechanism, the timeline we're hoping is going to be sped up dramatically compared to what we've seen in the past, where we'll be able to lump types of treatments together, and then we'll be able to move seamlessly from IND to phase 2 and 3 trials, and so get cures to patients much more quickly. But specific issues with applications, we hope that we, for products, we hope that we can also address in a timely manner, and I can't speak specifically to that application, but we're going to have the same standards of approval that we've had in the past, but hopefully be able to approve things more rapidly and in a more orderly manner than we've been doing previously with this plausible mechanism pathway. Thanks.
Thank you. We have Arthur right here. Thanks.
Thank you. Secretary Kennedy, Dr. Casey Means will sit before the Help Committee on Wednesday. What can Dr. Means do if Senate confirmed to move this innovation in rare disease therapies, and what do you expect from her? President Trump said you picked her as the Surgeon General nominee.
Well, we've been waiting for a long time for Dr. Means to come on board. We're very, very excited about her coming on board. She has an extraordinary capacity to communicate to the American public. That is the function of the Surgeon General. The Surgeon General is a moral authority for the public. They're a medical authority for the public on a whole wide range of issues. And we've been waiting for a long time for her presence. We're very excited about it. And I think hopefully she'll be on very, very soon.
Okay, right here on the end. Series? Yeah, that's fine.
Hi, my name is Miriam O'Day. I'm here today representing individuals who have alpha-1 antitrypsin deficiency. This is a rare form of COPD. I have a two-part question. Number one, in the policy statement that you listed online for plausible, it does not define what ultra-rare is. We know that rare is defined in statute as 200,000 or less. So will this policy be at the discretion of the FDA to be able to define what ultra-rare is? Secondly, with all of the innovative pathways that have been introduced over the years, they have all depended on urgent and unmet needs. And fortunately and unfortunately for individuals with alpha-1, there are 3 therapies in the marketplace that are available for them, and so flexible endpoints, clinical trial design, real-world evidence, these have not been used as biomarkers have not been used. CT densitometry, which could be used, have not been implemented in expediting review for alpha-1 antitrypsin deficiency therapies.
So it's a great question about alpha-1 antitrypsin, and when we were writing the draft guidance, we were very careful to leave it open-ended to not limit this to ultra-rare diseases, because it is an arbitrary cutoff between rare and ultra-rare diseases, but this could really apply to any diseases, even common, if they meet the criteria for the plausible mechanism pathway, the well-defined underlying etiology, a targeted treatment, and then evidence that the treatment addresses that problem and shows the benefit that we would see that's not consistent with the natural history of the disease. So, and in terms of real-world evidence, I actually just mentioned that approval that we had in CEDAR for Menkes disease, and that was actually based on real-world evidence, not a randomized clinical trial. And so when we're able to see that, as I said, the disease course is different with the treatment than what we would see with the natural history of the disease, that is really compelling evidence, especially when it's overall survival, though and we can also certainly look at other endpoints, which are also very important. So, Dr. McCary, yeah.
Isn't she amazing? We're aligned. You got to do what's common sense, and there's probably very few diseases that are a poster child for this issue, and I would put the alpha antitrypsin deficiency, and I've taken care of patients who have had it, at front and center in that underappreciated diagnoses with a whole host of medical implications. And if you look at a clinical endpoint, there's a number to pick from, but we got to use common sense. We have to use common sense. And that's what this day is all about. Can we use common sense? So the other point I wanted to add, and the reason I stepped up, is you also asked about real-world evidence. So in the past, when we came to the FDA, we learned a lot. One of the things we learned that was standard of care at the FDA is that to admit real-world evidence to the FDA, it had to meet a whole bunch of criteria. Requirements so onerous that only 12 drugs since 2011 had submitted any real-world evidence. And they'll tell you, because the requirements were so complex, you had to be able to, in a database, drill down to an individual patient's clinical data. Well, that basically means you need informed consent, and that means you can't use the SEER database, you can't use the Medicare database, can't use the Swedish national database, you can't use experiences that a physician has compiled over their experiences, multiple institutions. What are we doing? So we changed that. We announced our intent to change that guidance. You submit whatever you want. You submit any real-world evidence. If it's not good evidence, we'll let you know. But at least allow drug developers to submit the experience in the real world.
Okay, we'll take one more right there at the end. Yep, thank you.
Hi there, Maisha here from Inside Health Policy. Just a few follow-ups on the announcement. So who will conduct the review for these therapies, and is a separate review needed for each patient? And what kind of follow-up will FDA do after patients get the treatment?
All right, I'll take this one too. Very good question. So who is going to conduct the review? So this is going to be the reviewers in CBER and CEDAR who will conduct the review. And then could you state your second question one more time, just so I'm sure I answer it correctly? Yeah, so very good question. So the way that our career staff in CBER is looking at this now, based on a publication that came out in December in, let me just get the name of in American Journal of Human Genetics, and discussing with the review team there. We will likely be requiring separate INDs, but they will be under an umbrella protocol or application, and then we'll be able to take the INDs to phase 2 and phase 3 trials together. So it will kind of combine treatments that have a similar mechanism like genetic editing as described with baby KJ, but address different mutations that cause different diseases, but be considered really under the same protocol and clinical trial.
All right, thank you, everyone. We really appreciate you coming out, and safe travels home. Thank you. And then you all are gonna get together for a picture. We're gonna do a final picture of all you guys up here. So do you wanna get in front? Amy, you wanna tell them? I'm gonna get out of the way.